Hello, and welcome to this series of lectures on research ethics. And so what we're going to do this week in class is kind of start out with kind of an overview or introduction to research ethics, and then using a case study approach, look at a couple different questions that have come up relatively recently within the field of science, and apply some basic ethical principles to them, as well as kind of consideration kind of about what could be done better or could be done differently going into the future. As we've seen throughout in the course, science is a community. Science is dependent upon kind of an interaction with other scientists, and it's really built upon the work of others. And so individuals that are making really monumental scientific breakthroughs today are basing their work, at least in foundation, on the work of others. And that goes back throughout history. And so because science is so interdependent upon the work of others, we're really dependent upon individuals that are practicing science to have good ethics and good integrity, with the idea being that we want scientists to be impartial. We want them to be avoiding any potential biases that they may have, so that as they're doing experiments, designing the experiments, running the experiments, making observations, and then ultimately reporting the results of their experiments either in a scientific publication or in a presentation to the scientific community, we're really dependent upon those scientists acting in an ethical way and reporting their details, reporting their observations as accurately as possible, so that other individuals are able to look at their results and go on to the next step, to kind of think about things in a slightly different way, kind of expand on the work that's been done with by others, with maybe slightly different kind of modifications to the experimental techniques or the model system that we're working with, in order to be able to better understand kind of the phenomena that is going on around us in the world. Having said that, scientists don't have to agree with one another. They don't have to, completely accept any result that is published within a peer-reviewed paper. But the idea is that with kind of challenges to ideas, with kind of alternative viewpoints and alternative perspectives, it's dependent upon scientists to have a collegial discourse, to disagree respectfully, to talk about the potential limitations of a given study, maybe possible, take a look at alternative hypotheses, alternative explanations about what's going on. And through that, science is really a self-correcting process. And so an individual that may have, pretty good idea, but not so good in the execution, not so good in doing the experiment, can get the results out there, and it'll be, the ideas, the experiment, the observations can all be fine-tuned by other individuals working on it, and use that information to continually advance the field of science. Now, one of the most important things in science is the idea that you want the observations to be as accurate as possible. And so because of that, it's important for individuals that are doing experiments, that are making observations, to recognize if they have potential biases and the impact that their potential biases can have on both their design of the experiment as well as their collection of the results. And so because of that, when we do scientific research, we often build in mechanisms to kind of minimize the impact of biases on the experiment. And so in many experiments, they're described as being a single blind design. In a single blind design, the observer, the scientist, will likely know what different conditions, different test situations, different kind of groups that the subject may be in, but the subject is going to be naive. The subject is not going to know what it is that they're going to be necessarily exposed to, what are the expectations of the result. And so a good example of a single blind design study are the classic taste tests, where you'd have a situation, and this is kind of illustrated with coffee here, just because it's, coffee is an important part of science. We've got coffee from a variety of different vendors that provide coffee. And in a taste test, you wouldn't have the labels on, where the coffee is coming from, but you'd have an individual, the tester, that would potentially, have a blindfold or have anonymous kind of identified cups, and they would, sample the different kind of aspects of the coffee and be able to describe what they are. Again, this is going to be a single blind design because you don't have to have the situation where the subject is aware, what the different coffees are. An individual may prefer, Starbucks coffee, and so because of that, they're going to look at them. They're going to see the mermaid on the side of the coffee cup, and they're going to say, yeah, I like mermaids. I like Starbucks. Starbucks is my favorite, and so I'm going to rate Starbucks really well. But in a blind taste test, it's really dependent upon what is the subject experiencing, what is the flavor of coffee or the taste of coffee that they're looking at. So what we're doing within a single blind study, by having the individual not know what's going on, not knowing what group they're being in, or what material they're being tested with at that time, is that you're minimizing the subject's potential biases. But we need to keep in mind that the observer, the scientist doing this taste test in this study, is potentially biased, potentially towards the outcome that favors their hypothesis. And it's possible that in doing the experiment, they could be influencing their subject, handing them a coffee cup and saying, this one smells really good. You're going to like this one, for a coffee type that they're, kind of leaning towards. And, ooh, what's in here? As they pass over a coffee cup of something that they don't like. And those kind of actions, either intentionally or unintentionally, can have an effect in influencing the subject. There's also the possibility within a single blind study that the observer could be biased in recording the results. So if the observer says, I'm not sure, it's kind of good, kind of bad, but the observer, the scientist doing the experiment, wants to see a good result, they may kind of record the kind of good, kind of bad as more towards the good kind of aspect of the recording aspect. And so therefore, potential biases are an important consideration when you're taking a look at any experimental design, but especially when you're doing a single blind design. Now, one of the important reasons why this is a consideration within science is a concept known as the Pygmalion effect. And the Pygmalion effect has been looked at within psychology and has been given the term the Rosenthal effect. But basically what you have with the Rosenthal effect or Pygmalion effect is the idea that the scientist or the observer's expectations are going to affect the other person's performance. And so kind of scientific, psychological explanation of the Rosenthal effect is that it's an observer expectancy effect. But basically, you run up into the situation where you can generate a self-fulfilling prophecy. And so the idea is that, you start here within this diagram, within the circle, that, the observer's belief, I expect people to do well in this situation. And that's going to potentially influence our actions towards the others. And so if I expect them to do well, I'm going to be confident in interacting with them, and I'm going to convey that confidence to others. And by doing that, it has an impact on the other person's belief. And so this would be the subject over here. So if we're very confident in the ability of the subject to do this, we're going to, in our actions, again, either intentionally or unintentionally, kind of influence the actions of the subject, the person that we're looking at. And by doing that, it kind of alters their action so that they're going to be doing better on the test or performing better in some way. And because they're performing better, that's going to reinforce our belief that they're going to do better on this. And that, again, is going to kind of influence them through our actions, having an impact on them. And so this kind of continues in this cycle. And so examples of the Rosenthal effect or the Pygmalion effect would be seen in the, again, as I said, the self-fulfilling prophecy idea. If you have high expectations of individuals, this is going to tend to lead them to have better performance. If you have lower expectations, potentially it would change our way that we interact with the individual and has the potential to cause a worsening in their performance. And so examples of that would be, examples of stereotype biases or the placebo effect. So again, because in this case, in a single blind study, the observer is aware of the situation, is aware of what they expect the subject to be doing, they could potentially be influencing them and changing their behavior. Now, a good example of this is the placebo effect. Now, a placebo is an inert substance or inert treatment that's often used as a control because it has no therapeutic value or has no impact on the outcome that we're going to be looking at. And so examples of this kind of in a medical setting would be providing like a sugar pill or a salt pill to an individual. That individual then thinks that they're getting this miracle drug, this miracle cure, and so they have a better mindset, they have a better outlook on life, they may be following the instructions of the doctor a little bit more precisely than somebody that has a poor outlook. Within an experimental setting, what you could be looking at is like a sham surgery or a sham procedure, where you're basically going through doing everything except for adding in the treatment effect. Now, the placebo effect is fairly controversial. There's some studies that show that there is a placebo effect. There are other studies that show that it's a result of the bias either of the observer or a potential bias of the response of the subject. But regardless, there's some good evidence that there is, an effect by the Rosenthal effect, by the Pygmalion effect. And so it's not beyond reason that the placebo effect would potentially have an altering effect on an individual. If you think about it, you give somebody a sugar pill and said, this is going to make you feel a whole lot better. Chances are they're going to feel a little bit better just from the sake, just from the instance of taking the pill, the sugar pill in this case, and listening to the doctor's instructions that this will make them feel better. So the ideal situation in many experimental settings when you're concerned about bias is to undergo what's referred to as a double-blind design. Now, in a double-blind design, neither the observer nor the subject know the specific conditions that the subject is going to be exposed to. And so many clinical trials are done in a double-blind design. And so patients will be recruited or subjects will be recruited based on specific criteria. But then they're going to be randomly assigned to either a treatment group or control group. So they'll be assigned to a group. They may get, kind of a collection of pills. If we're looking at a clinical study like the illustrated on the diagram over here. They get a collection of pills, but they're not quite sure what are in the pills. It could be a control situation, or it could be a test situation. Now, the doctor just gets the box of pills. They aren't aware of what's within the capsules themselves. So they can't influence the subject based on their interaction with the subject. You're getting, lucky you're in the test group, you're going to get the experimental drug and you're going to do really well. Or, well, I'm sorry, you're in the control group. It's good for science, but probably you're not going to see much of an effect. That could potentially bias both the doctor as well as the subject in how they're going to be looking at their effects within the experiment. And so with the double-blind study, the doctor doesn't know about it, so they can't be influencing the subject. And they're also not going to be subjective when they're reporting. And so, if it's a questionable observation, They may lean towards positive if they're looking at a test drug, and lean towards negative if they're looking at a control placebo group over here. And so again, that double-blind study eliminates the bias. And again, there's potential bias within the individual as well. If you let the individual know that, this should be having an effect to them, especially in subjective reporting, pain tests, on a scale of 1 to 10, what is your pain? If you're given something that you're told is going to alleviate the pain, there's going to be a tendency of subjects to report less pain than what they're observing. And if they're in a situation where, I'm sorry, we can't do anything about the pain, it's only going to get worse, people are going to tend to, give a higher pain score when they're asked about it. And so that's an example of experimental design and potential impact of bias, both of the subject and of the observer. But there are other situations where we also need to think about what we're doing and the potential effect of what we're doing in kind of reporting the data as accurately as possible. Okay, so in the second example, what we're going to look at is what do you do with data outliers? So we're going to set up an experiment. We're going to have 2 groups. We're going to run 7 repeats or 7 trials, and we're going to record the results. And so we've got group 1 in the first column, group 2 in the second column. We're going to run a t-test comparing group 1 to group 2, and we get a p-value of 0.993. So using what we know about kind of experimental design and inferential statistics, we take a look at this. P equal 0.933. P is greater than 0.05, so we're going to accept the null hypothesis and basically say that there's no difference between the groups. There's no difference between group 1 and group 2. And we're really disappointed because we were really hoping that group 2 would be doing better than group 1. And so we start looking at the results and we say, okay, in trial 1, group 2 is about a point better, half a point better in trial 2, 1.5 better, 1.5 better, 1.5 better, point better. So, 1 through 6, group 2 is always doing better than group 1. Now, all of a sudden in trial 7, group 1 is 18 and group 2 is 12. That's strange because in the trials 1 through 6, group 2 was always higher than group 1. And now all of a sudden in this example, group 1 is higher than group 2. So that's different. If we take a look at the values, we see that we've got a range from about 6 to a range of about 9.5 in trials 1 through 9. And all of a sudden in trial 7, we got, values of 12 and 18, a whole lot larger than what we've got here. So if we take a look at this and we say, okay, the results of trial number 7 are very, very different than our trials 1 through 6. So, maybe it's trial 7 that's screwing us up. Maybe trial 7 is interfering with our experiment. And so we take a look at this and say, okay, something went wrong with trial 7. I'm not sure what went wrong, but something went wrong. The results aren't right. It doesn't fit with trials 1 through 6. And so something went wrong. I don't know what with trial number 7. I don't want to admit I made a mistake in the lab. I don't want to have to admit to my peers. I don't want to have to admit to, the PI, the principal investigator, the mentor, the person running the lab. So I'm going to look at this and say, okay, it just, 7's an outlier. The results are just wrong. I don't know why, but they just don't look right. And so we're going to drop them out. And so, okay, just striking them out and running the statistics again, all of a sudden, by omitting trial 7 and running trials 1 through 6, we get a p-value of 0.043. And there was much rejoicing because p is less than 0.05. Therefore, we can reject the null hypothesis. P less than 0.05, reject the null hypothesis, which tells us there is a difference between groups. So there's a difference between group 2 and group 1. And we look at it, and group 2 is higher than group 1. So whatever it is, group 2 is doing better, or the treatment on group 2 had an effect to improve the performance of the outcomes in group 2. And so, this is great. We did the experiment, had to toss a little bit of an experiment out, but we got a p less than 0.05. We got lots of things we can talk about in our paper. But what we've done here, because we threw out this data without having a really good reason, is really data falsification. And so in the book by NIH on Being a Scientist, A Guide to Responsible Conduct and Research, data fabrication is referred to as manipulating research materials, equipment, or processes, or, and this is the important part for this one, changing or omitting data or results such that the research is not accurately represented in the research record. And so what we've done here by omitting trial 7 without a reason, other than we didn't like the results, is we've gone into the concept of data falsification. So we've basically done something unethical with our results. But let's look at this example a little bit differently. Let's say we took notes along the experiment, and I'm not really going to worry about the notes in trials 1 through 6, just because I ran out of space on the slide, but we're going to assume that the notes on 1 to 6 said, experiment went well, everything's going good, time was right, equipment went well, labels were great, everything was fine. But then we checked the notes on trial 7, which we said, had different results, looked differently from what happened before. And we look at the notes. And so in trial 7, in the notes, the fire alarm went off during the experiment, so we had to evacuate the lab. When we returned to the lab, the test tubes had been knocked over and the labels had fallen off. The labels were on the table. The tubes were there. I think I put them on in the right place. But I'm not quite sure, but they're probably right, maybe wrong, I'm not quite sure. But because we had to evacuate the lab and come back and then relabel everything, the overall experiment much longer than previous trials, which maybe is why the numbers are so high. So we had lots of things going wrong with trial number 7. And so we see the conditions for trial 7 were different from the previous trials. Previous trials were fine, no fire alarm going off, no evacuation, the test tube stayed labeled, we were staying on kind of our timetable, we moved things when the alarm went off, everything was fine. So we take a look at it. 7, we just had a lot of problems with trial number 7, as well as the possibility that the tubes were being mislabeled. Now at that point, you don't say, okay, now I have the justification to strike this data, so I'm going to cross it out now. What you need to do is really discuss the situation with your research mentor. Talk about this with, the person in charge of the lab. Not, in the previous example, I don't want to admit making a mistake. In science, we got to live up to it. We got to say, okay, not quite sure what happened here. Here's a possible explanation. What do you think? Discuss it with your research mentor, and the decision was made jointly that we could omit the results because it's unlikely that trial number 7 was conducted in the same ways as trial number 1 through 6. So we might be able to strike it, or it might just be that, okay, we're going to completely redo trial number 7, so we get 7 trials. But we have a reason, and we've discussed that reason with somebody else. So we've eliminated any potential bias, hopefully, by doing this. If we're biased and the mentor is biased, we need to have a conscious decision about whether or not it's okay to strike this data or to repeat this experiment. But in this case, we're going to assume that there are good reasons for omitting the results from trial number 7. We can justify it based on the situations being different. Then when you run the statistics on trial 1 through 6, we get a p.043, p less than 0.05, reject the null hypothesis if there's a difference between the groups. And so in this case, we don't have the data falsification that we had in the previous, where we just struck it just because we didn't like it. In this case, we omitted it with a reason that is justified and can be explained to others. Then we can go through and take a look at the data. Now, another example of this is this idea that you can go through, especially with desktop publishing, Adobe, can't remember the name of the Adobe program, but the Adobe program for drawing stuff, Photoshop, I think is what it's called, or any of the drawing programs that you can get on your computer. You can go through and really change the contrast or change the appearance of slides. And so in this one, I just pulled a simple Western blot off of the web. This is what we've got over here, but we really expected to have a 37 kilodalton band. I think that's 37 kilodaltons there. We expected a band here in lane 5. And we look at it, and it's, kind of there. It's probably there. But I really want to show that, in my publication. And so I'm just going to draw that in, make it more prominent, so that, it's there. I know it's there. I'm just going to make it very clear to an observer that's looking at this figure in my paper or my poster or some other time. Now, in this case, we've really altered our Western blot here. We've gone through and made up data or made up results. And in that case, what we're looking at is an example of what's referred to as data fabrication. So we're making up data or results. And so this is unethical. But there are times in which you're going to run a blot, and it's not exactly the same as, what you're seeing when you're looking at it in real life. You're taking a photo of it, the photo's not showing it, as well as you would like. So you can go through, and I don't have the Adobe Photoshop with the ability to adjust contrast and brightness on my computer. So you get a kind of an exaggerated version of it. So you got the original photograph here. It's fine to go through and change, like the brightness and the contrast of the entire image. In this case, making everything a little bit darker. So you still see, these are really dark bands. These are really lighter bands. And then, on lane 5, when we're trying to look at it, maybe a little bit darker there, but it's still kind of in relationship to all of the other bands here. So this, within reason, is allowable. You're going through, you're changing the brightness and the contrast of the entire image without kind of selectively enhancing a region that you wanted to kind of reinforce like we did over here. So again, that's something that's allowable, but again, it's something that you should be discussing with your research mentor or the other people within the laboratory. Okay, in the next example, we're going to say that what we want to do is go through and count cells. And so we got a picture here of human embryonic stem cells and cultures from UCLA, again, stolen blatantly from a website. So we go through this, we're treated in some way, and what we're interested in is counting the cells and trying to get an idea about how many cells are growing under this condition. And we're going to compare it to cells growing on another condition. And so we go through, our hypothesis is that the culture conditions are going to discourage growth. As we know, in kind of the research design, we're going to measure or count the number of cells in 5 squares of a consistent size within this field of view. And so we're going to look at it, and my hypothesis is that it's going to discourage cell growth. So I'm going to look and I'm going to count squares, again, these grid patterns, which have no cells in it. And again, we've selected this, based on our hypothesis, and I'm making an over-exaggeration here, but you kind of get the point. In this case, we got like 0 cells in all of these. And so we could say, okay, we measured 5 squares. We had 0 cells in those squares, and therefore these conditions discourage growth. The alternative is that hypothesis 2, the culture conditions promote cell growth. And so if we're going to do that, we're going to find really interesting squares. And we're going to take the same 5 grids, but instead of looking at them in areas where we don't have any cells growing, we're going to look at areas where we've got lots of cell growings. And again, we've, based on our hypothesis, biased our observations. And again, keep in mind that we're not going to go through and count all of the cells within this entire field. We're going to sample it. And so we're going to ideally sample different regions of it and use that as a description that would be kind of indicating what our results like without having to count all of the cells in the screen. We count them within these 5 squares. But again, in both of these cases, we were biased, again, exaggerated, but biased based on our hypothesis for where we put the squares. And so a better way to do this is to set the criteria for our observations before collecting our observations themselves. And so the idea is we're going to put, and it's referred to as a reticle, but we're going to put kind of a grid-like pattern into the eyepiece of our microscope. And we know that we're going to count this square, this square, this square, this square, and this square. So we've still got the 5 squares. There's still kind of the random, or not random, they're all of the consistent size, and they're going to be located kind of in a preset position. And then we're going to bring in the image, and we're going to say, okay, we're going to count the cells within these pre-selected grids and use that to describe our overall results. And so when we do that, we see, you know, a couple squares, we've got a lot of cells, a couple squares, we have fewer cells, one square that's kind of in the middle. And so therefore, you know, if we take a look at these 3 different examples, we've gone from 0 cells to a lot of cells to, you know, probably what we've got here. So kind of a mixture, kind of a medium number of cells, which are going to be present in that. So again, by setting up the criteria ahead of time, where we're going to count our cells within this grid pattern, we're eliminating the potential for bias. And then what I also like to talk about, again, is how reliable our observation is going to be. The idea is that we want science to be reproducible. We want science to be able to be done by other researchers. And if they were to do the experiment in exactly the same way, their results are going to be consistent with the results that you're going to be reporting. And so there are 2 ways to test a reliability of an observation. The first way is to take a look at intra-rater, I-N-T-R-A, rater reliability, is how consistent an observer is at recording the same phenomena in the same way. And so in this example, looking at a Rorschach ink block image, and we're going to say that, you know, I take a look at this image as I'm lecturing in Bio 102, and we're doing insects. And so as I prepare my Bio 102 lecture on insects, I'm going to look at this and say, okay, you know, that kind of looks like a moth. And so look at that. Yeah, here's a Sphinx moth. It looks, you know, not exactly the same, but it's kind of the same. And so that could be, you know, take a look at this and we got a moth. But instead of, you know, I'm going to go back and I'm going to look at this a couple weeks later when we're taking a look at mammals and we're taking a look at bats. And I'm going to say, okay, I'm preparing my Bio 102 lectures. I'm kind of biased towards mammals and bats right now because that's what I'm researching and putting into my lectures. And I'm going to look at this and say, okay, in this situation, a couple weeks ago, I looked at this and thought I saw a moth. Now I'm going to look at it and I think I'm going to see a bat. And now, you know, 2-3 weeks later, we're, you know, broadcasting from home. I'm preparing a D&D campaign for my son to do, putting together a bunch of things. And now I'm thinking about demons and dragons and all kinds of things. I'm going to look at this and say, okay, what I thought about in February as being a moth, what I thought about in early March as being a bat. Now, as we go into April, now I'm thinking it's actually a demon. So in this situation, again, exaggerated, my intra-rater reliability isn't so good because I looked at the same object, the same phenomena, and depending upon when I looked at it and what was going on at the time, I recorded it very, very differently. And so that's going to be important, again, when you're making observations within the laboratory, is that you have good, ideally objective criteria that you can look at and ideally use that information to consistently report in the same way all throughout your experiment. And so that's intra-rater reliability. So within the same observer, looking at the phenomena multiple times and getting the same result. The other thing to look at is inter-rater reliability. And this is especially important when you're doing like clinical studies where you're looking at results maybe of a drug test at multiple sites, or you're doing a physical therapy intervention and you've got a lot of different physical therapists that are working with a lot of different patients at a lot of different locations. What you want to have is good inter-rater reliability. Inter, I-N-T-E-R, inter-rater reliability is how consistent are multiple observers at recording the same phenomena in the same way. And so in this case, you know, you would train the observers on the criteria that you're going to be using with the hope that everybody's going to be looking at things the same way. again, exaggerated within this, this cartoon over here from the poem by John Godfrey Saxe, Blind Men and the Elephant. Basically, we got scientists coming up representing the blind men, so probably wearing masks or blindfolds. And depending upon where they are in relationship to the elephant, they're going to feel it. So the guy feeling the tusk says it's a spear. Guy over here taking a look at the legs, saying it's a tree. Actually, that looks like a girl, female scientist. A rope back here with the tail, a wall for the body, the ears over here potentially as a fan, the trunk as a snake. So again, all looking at the same phenomena, all looking at the same object, but based on where they are and based on their perspective, they're recording it as very, very different. And so again, when you're designing a study, what you want to do is make sure you have good criteria so that you make the same observation multiple times and record it the same way. Or if you've got multiple observers, you have, again, good criteria, good training, so that they're able to look at the same observation and record it consistently between people. And again, that's going to be important as you're collecting results that are ideally going to be reflecting what is actually occurring within your experiment. And then finally, I want to talk about other potential biases. As you've read the scientific papers this semester, you take a look at the acknowledgments. What you're often going to see is that the scientists and authors are going to disclose where's the funding for the study coming from, who was involved with the study, what was their role within the study, and ideally identify if they have potential conflicts of interest. And so conflict of interest are things that could either intentionally or unintentionally generate biases within an individual. So examples of conflict of interest would be, employment, business interests, financial investments. So if you have bought into a drug company that is testing a new drug, and you now are going to test it in a laboratory, if you come out with really strong results that say this is a miracle drug, it's going to cure, whatever it is that it needs to cure, you're potentially going to get a benefit because, your stock options or your investment in the company are going to become more valuable if the drug works. In many cases, industry grants or industry sponsorship could potentially cause biases of an individual. And therefore, individuals that have received funding from, say, pharmaceutical companies or corporate sponsorships will disclose that. It doesn't mean that they have to be kind of ignored in their work. It doesn't mean that they can't do the work, but it means that they've identified that they could be perceived as having a conflict of interest, put it out there so that people are able to look at their results and their conclusions in the relationship to that and make a decision. Other biases, could be personal considerations, pet theories. We talked about a couple of examples where, you had your hypothesis going in and you kind of altered your results or altered your kind of way of recording the results to support your pet theory, your pet hypothesis. And so basically what I'm hoping you see in this series of lectures, as well as this first lecture on research ethics, is that it's really important that you're aware of any potential biases you have as you're designing experiments, as you're collecting results, and as you're reporting results. Because in science, we are dependent upon people being as objective as possible, as impartial as possible, so that they're going to be accurately reporting their results, so that we can then take their results as an accurate representation of what they observed within the laboratory and use that as a foundation for our own work, again, because science builds upon the work of others. Thank you. We've got a good collection of case studies that I think will be hopefully interesting to me, hopefully interesting to you, as you take a look at different aspects of scientific ethics and research ethics. Thank you and have a great day.